Good morning, everyone. I am Giorgia Cassandro. I'm a project manager of CORIS, and today I'm here in lieu of Professor Gerosa, who had an emergency at work and so is not going to be present. As you probably know, today is the third day of the winter school organized by CORIS and Genome Quebec, with the aim to seek the collaboration between researchers from Italy and from Canada. The topic of today is artificial intelligence applied to genomics, and we have the opportunity to discuss with some of the most important researchers from University of Padua and from McGill University and McGill University. And now I leave the floor to Stéphanie Lafontaine, that is the Vice President of Genome Quebec.
Thank you very much, Georgia, and thanks everybody for being here today. So good morning or good afternoon, whether you are in Quebec or Italy. So as Georgia just said, I'm Stéphanie Lafontaine. I'm the VP of Scientific Affairs at Genome Quebec. Just briefly, Genome Quebec is a not-for-profit organization with a mission to catalyze the development and excellence of genomics research and accelerate the discovery of new applications for genomic and health. And Quebec is really a world leader in artificial intelligence research, and together with genomics, we really believe that it will become an important driver for precision health applications. So AI is one of Genome Quebec's strategic research areas, where we recently funded over $3 million for health research project, and I'm really glad today to see that some of our funded researchers are panelists today on this panel. Also, before we start, I would also like to take the opportunity to thank our co-organizer on the Quebec side, Fonds de Recherche du Québec en Santé, which is the Quebec funding agency for health research. Its mission is to promote and provide financial support for all areas of health research, to support graduate students and training careers of researchers, and fund research centers and thematic networks. So briefly, very happy to be here today and looking forward to the panel discussion with the hope that it will be the basis of new ideas and future collaboration. Thank you.
Thank you, Stéphanie. And now I leave the floor to Professor Leonardo Salviate, that is a professor of genetics in University of Padua, that will be the moderator of this roundtable.
Thank you very much. I also want to thank Professor Gerosa for having me here today. And the issue of artificial intelligence to genomics is one of the hottest ones in the field, because genomics is an extremely wide field of research, which includes which spans from clinical genomics, so the analysis meant to identify the causes of genetic disease in individual patients, to drug discovery, gene expression studies, development, and whatever. And the novel machines, the novel techniques, literally flood us with data, and it is becoming more and more challenging to have tools to interpret this data, because the human mind simply cannot grasp the complexity of what these analyses are telling us, of the numbers and of the variants that they are providing us. So I think it's a very interesting occasion here, because we have people with different expertise. I see people coming from mathematics, biostatistics, engineering, and also from the wet sciences field, and to have them together in a single workshop, I think it's important to share these. Sorry, it's the lab calling me. I will have eventually to answer, but because we have prenatal diagnosis going on, and so to have them all these different the expertise here, it is important, because the multidisciplinarity is becoming a critical issue to tackle these problems. And so, having said this, the first speaker today is Professor Mauro Conti, who is Professor of Cybersecurity here at the University of Padua. And he will start with presenting his research and his interest in this field. Please, Professor Conti.
Good afternoon, everybody. I think you can see my slides, right?
Yes.
Okay.
Yes.
So thanks, Professor Salviate for leading this panel. Let me just briefly also thank the organizer of this panel, this event, Professor Gerosa. Thanks also for substituting and leading the presentation in the beginning. So I will start giving my self-presentation very briefly. So I will try not to annoy on what exactly has happened in my academic career. You can see my main affiliation in this slide. As said, I'm currently a full professor in computer science, and the area of expertise is cybersecurity. So that's very orthogonal to some of the things that we are going to discuss in the core topic of this workshop. Again, I'm not do strictly speaking with genomics, but as we will see, there is actually a strong intersection of interest with this topic. You can see also the fellowship that I received. Again, most of them I received in the specific area, of course, of computer science and cybersecurity. So what are my research area of interest? Just to give you a broad idea, and then maybe we can focus on why should I be interested in genomics. So I work in all different areas around security, cybersecurity, and privacy. Just to give you an idea, I did several works in mobile computing, like Android is one of the specific operating system, but in general on mobile computing. I do security in network security, in traditional network, future internet architecture. I do works in a side and covert channel to identify possible leak of information. In this case, there is also significant application of machine learning. One of the example is where we noise of the key press, we try to infer what somebody is typing on a keyboard, like it could happen in this conference call if you don't unmute your microphone. And then we work on technologies like social networks, IoT, cyber physical systems, which are all the systems which are composed both of cyber and physical components. And somehow, I mean, DNA and genomics might be around this topic, but in the past we mostly worked in automotive security, drone security, industrial control system security, and last but not least, we recently also working a lot on adversarial machine learning. Adversarial learning is this area that tries to understand the vulnerabilities of the machine learning systems. Here we have one very famous example, which is used in image recognition, where, for example, adding some pattern, which is basically non-influent for a classification. So I think all of us, we would agree that the picture on the right is still a panda, but automatic classifier would evidently fail in classifying this as a gibbon. So this is just one example. So why I care about the topic of this event? I care where the technology comes in. As we know now, there is. I found this article. I remember it's quite old, 2012, and nowadays we can do a whole genomic sequence in a small device that can be attached to a smartphone. And nowadays, our smartphone can probably easily store even the whole genomic sequence. And this, as you can imagine, opens up a very new set of applications that were, of course, unthinkable or decades ago. Here we have just some example. Of course, you can easily do when you have the whole genomics paternity test. You can do genetic compatibility, and maybe this will be soon available in a new Tinder version. And of course, very important, you can do personalized medicine. But in all this type of application and future one that might come up, you need to share data. And of course, I mean, if you want to do one of these tests, you do not necessarily want to share all your private data with the other party. So this is where the cybersecurity and confidential computation comes in. Why is this important? Because of course, I mean, I think it's very what is the difference from traditional security, traditional credentials that we have to our more private information, which is the DNA. Because if we lose information on Facebook, like it happened already on Facebook and on several other systems, of course, your account is at risk. Your Facebook chat is a risk. Whatever you do in the chat, I mean, your private things, affairs, or whatever business you want to do might be exposed. But imagine what is the exposition if DNA is lost. You don't lose only a few messages, you do lose your privacy, and also, most importantly, you also put at risk also the privacy of relatives, not just yourself or your chat, but you put at risk very private information about yourself and your relatives. So this is where, I mean, my expertise and interest come in securing the data and the computation over data. Of course, there are techniques like private set intersection, secure multi-party computation that allows this, but there is a new, let's say, specific research area. So these are some of my publications in this subdomain that involves DNA and genomics and security. And just a disclaimer for the continuation of my discussion later on. As I said, I'm mostly computer scientist and a cybersecurity expert, but I'm not none of the following. I mean, I'm not a physician, biologist, bioengineer, genomic expert. So if I do some not very correct claim from the genetic view, please bear with me. Thank you.
Ecco qua, sorry, I couldn't switch on the microphone. Thank you, Professor Conti. Your genetics was good. You didn't say anything that.
So far.
No, the reason, there's one thing that I will come up very often today, that the fact that I think that we are overrating our genetics, knowledge in genetics, because we still don't know how to classify mutation variants in the cystic fibrosis genes that has been discovered years ago. So let alone the complexity of several million single nucleotide variants that interact with each other. So thank you very much for your presentation, and please now I want to give the word to Professor Amin Emad, who is assistant professor at the McGill University in Toronto. No, in Quebec. Where is McGill? Toronto, no. Where is
Montreal.
Montreal. Then, sorry, my geography used to be good.
No worries. Thank you very much for the introduction, and thank you for the organizers to invite me for this exciting panel. I'm hoping that my internet connectivity is okay. And
yes,
but if the, okay,
we can hear you and see the data perfectly, so don't worry.
Okay. Very good. So good morning, everyone, and I imagine good afternoon in Italy. Today I will be talking, my name is Amin Emad. I'm an assistant professor at McGill and Mila Quebec AI Institute. Today I'm going to talk about machine learning in precision cancer medicine and some of the recent projects that we have been working on in the past few years, and I'm quite excited to share some of the high-level results and observations and to share with you, and also some of the challenges that I think are quite important to be aware of in the field. So as we all know, cancer is a major health concern around the world, and precision cancer medicine has promised to improve the outcome of cancer patients. And the idea there is we want to identify the best course of treatment based on different characteristics of the patient and their tumor, including clinical variables and molecular omics profiles of their tumors. However, in spite of various successes in this area, there are still challenges to be addressed. Particularly, two important problems are what are the molecular mechanisms that influence the sensitivity of cancer cells to a drug, and also what, if given the profile, given the characteristics of a tumor or a cancer cell line, are we able to predict whether they respond well to a specific drug or not before any drug was given. So in this talk, I will talk about this idea of drug response, which is a measure of sensitivity to a specific drug, but depending on the context, it may mean different things. For example, if you're talking about the tumor, it could mean that after the drug was given, did the size of the tumor get reduced or not? Or if you're talking about cell lines, we may mean that, for example, after the drug was given, what dosage of the drug was necessary to kill half of the cancerous cells after a certain amount of time, let's say 48 hours. Okay, so the first problem that I was interested in was, are we able to take the gene expression profile, basically the mRNA expression of cancer cell lines before any drug was given, and also the response of all of those cancer cell lines, and determine what genes are important in determining the sensitivity of these cancer cells to a specific drug. Why is this important? Because if we know, for example, a gene that is overexpressed in a specific case before the drug was given, determines the, or underexpressed, determines the sensitivity to the drug, then that could be a potential new target to overcome resistance to the drug, which is a major problem in cancer. So from the machine learning perspective, we can think of this as a classical feature selection analysis, and even simple correlation is widely used for this task. So the setup is that we have a matrix, a feature matrix like this, when rows are cell lines, columns are genes, and we also, for a specific drug, know some continuous value that shows whether the cell line was sensitive or resistant to the drug. And we can do, for example, correlation analysis or other type of feature selection, but the problem to remember is that we are working in a domain that the number of features is much larger than the number of cancer cell lines, 10 times, 20 times if we are lucky. So there is a high chance of overfitting or high chance of getting false positives. So how can we improve this? The idea was that given that we know different genes in a cell interact with each other to influence drug sensitivity, can we take advantage of a priori annotated protein-protein interaction networks in order to improve the prediction of correct genes? So I developed a method called progeny. If you're interested in the details, you can find it here, published in Genobiology, where we are using graph representation learning, using random walk with restarts, where the idea is that, kind of intuition of it is that if a gene is associated with the drug response itself, but none of its network neighbors are showing any type of association, that may show that there is a lower chance that it's actually a true positive. But if a gene is highly associated, but its network neighbors in a broad sense are also associated, it may be a sign that it's actually a true positive. So given this approach, we can obtain ranking of genes for different drugs and obtain the top genes, but how can we validate our results? So we looked at two approaches. One of them was a computational way where we selected the top identified genes using different gene prioritization methods, and then we can train a machine learning model only on those genes, and then see how well we do on held out data. And we showed that compared to many different methods, this approach gives us better predictors for this task. But of course, this is only an indirect way of assessing how well we are doing. So for 3 of the drugs, we selected the top 15 genes identified using progeny, and our collaborators at Mayo Clinic performed some gene knockdown experiments in cancer cell lines, and we found out that 73% of the genes that we had identified are in fact true positives, and their knockdown significantly changed the response to these drugs. So this was a good starting point, but we were also wondering, can machine learning help us to just, instead of only finding the top genes, predict the drug sensitivity of cancer cell lines, and how can we improve on currently existing methods. So for this purpose, we developed another method called Big DRP. You can find it currently, it's in bioRxiv, but it's under review, where we are integrating different data modalities, the chemical structure of the drugs, the gene expression of cancer cell lines, but also a bipartite graph that connects drugs to most sensitive and most resistant cancer cell lines. And we are using a heterogeneous graph convolutional network to integrate these different data modalities to get representations that can improve the prediction performance. And we showed that it actually does predict better. But maybe more exciting for us was that this has helped us to identify two novel drug combinations that show very cancer-specific synergy and we tested them. Our collaborators and McGill tested them in vitro, and we have identified that our predictions were truly correct. And we are currently working on follow-up on the mechanisms of these drug combinations. So now talking about drug combinations, that's also a very important problem in this domain, because nowadays the standard of care involves multiple drugs being administered simultaneously. And we would like to be able to also predict the response to drug combinations, basically whether two drugs in a specific cancer type are synergistic or antagonistic. Do they work well together or not? But the problem from a computational perspective is quite challenging, because the space of these drug combinations is very large. And to have data to train any machine learning model, we rely on experiments that are expensive. So even if we go after a database, the largest database currently available, we would get a situation like this. So for a specific cancer cell line, this shows the sparsity, how sparse the data that we have is. So we have more than 4,000 unique drugs, but then when we actually look at how these pairs of drugs have been tested experimentally together, we see that only at the very corner they have been tested and we have data on it. And then thinking that there is another third dimension in this three-dimensional tensor that we are not showing here, which shows different cancer cell lines, and they may have a different sparsity pattern. This shows how challenging this problem is from a computational perspective. So even completing this matrix can have a significant impact in this domain. So for this purpose, we have developed a method called MARC, which defines this as a multitask learning problem, where we are predicting not only the single drug response, but also the synergy scores of the drug pairs. And we're working on preparing the paper at this point. Now, up to this point, we have been focused on preclinical models of cancer cell lines. But the question that at the end of the day we care about is that how would actual patients, can we predict on actual patients and whether they are responsive or sensitive? Now, ideally, in a traditional machine learning setup, our goal is that we have a training set that is representative of our test set. So the best scenario for us would have been to train our machine learning models on cancer patients and then predict on new cancer patients. The problem is that the data on cancer patients for new drugs, maybe drugs that are being going through approval through clinical trial, or they may be even newer than that, is unavailable. So it's not easy to be able to train models. And even for the ones that we have, still the data is quite small. So one idea would be that can we train machine learning models on preclinical cancer cell lines and then somehow adapt them in order to be applicable to actual patients. The problem, however, is that the distribution of the data from these different domains, preclinical samples and clinical tumors, are quite different. And there are different reasons for it. For example, different labs are involved, potentially different biological protocols for sampling, or even different technologies for measuring the omics profiles. But also, there are biological reasons for this, where cancer cell lines are not the best models and not the best representations of real tumors. So there are significant differences between these two domains. And we were wondering whether the models that have been already proposed in the literature actually can predict on real cancer patients. So we tried many different methods that have been proposed. And we observed that most of them do not do a good job on being working on actual cancer patients. And the only thing that we found out that actually works relatively better was a method that we developed called tissue-guided lasso, where we are including tissue information from cancer cell lines and tumors in a non-traditional way. And even then, when I say improvement, we were doing a good job only on 60% of the drugs that we have tested. So still there is a lot, there is a lot of room for improvement in that area. So after that, we looked at different approaches for domain adaptation and particularly deep learning models. And we developed a method called Tyndall. If you're interested in the details, you can find it here. And now we are doing a good job for more than 70% of the drugs, but still we are hoping to do better in the future. But in addition to that, we used explainable AI techniques to make this deep learning model more interpretable and identify genes that are actually contributing to the prediction for the drugs that we are doing a good job. And we selected the top 10 genes for one of these drugs, tamoxifen, and our collaborators again did some gene knockdown experiments. And for the first time, we found 7 novel genes that in fact, as we predicted, significantly influenced the drug response as potential novel targets. So this was some summary of some of the problems we are interested in, but I just wanted to also mention some of the major remaining questions that exist. One of the most important ones is this idea of domain discrepancy between data sets that we have available. This is not just in pharmacogenomics, but in many different bioinformatics areas. It's a major problem. Whenever there are different labs, different protocols, different technologies are involved, or different models of a specific disease are involved, when you actually look at the distribution of data, they do not match in a way that allow us to do good machine learning. And solving this issue and dealing with this domain discrepancy is a major problem, and it's something that is very interesting and exciting to solve. Another thing that we are interested in is getting better representation for the chemical structure of the drugs, particularly for designing novel drugs. That's another area that we are quite interested in. We are also working on better understanding transcriptional regulatory networks that are involved in drug response, for example, transcription factors that influence genes that influence the drug response, and also mechanisms of RNA alternative splicing that can help us better understand what happens with regards to the drug response. So finally, I would like to acknowledge our collaborators at McGill and Mayo Clinic, as well as various funders that without their help, none of these projects were possible. Thank you very much.
Thank you very much for your beautiful presentation. And there are many questions that your results raise in terms also of the transition to clinical medicine, because the ideal situation would be have to just have a sample of the tumor, put it into some even a sequencer or whatever, and to have a report saying you have to give this drug with this dosage for this time. This is, I think, the ultimate goal of research. Anyway, questions later. At this point, I have the privilege to give the word to Professor Dario Gregori, who is Professor of Biostatistics here at the University of Padua. Please, Professor Gregori.
Thank you.
See you. We see a fixed image of you. And now we see you moving. Okay.
Okay, wonderful. So I share a few slides. Okay. So thank you, Professor Salviate, and let me thank you, the organizers, Professor Gerosa and Genome Quebec, and also a special thanks to Georgia for all during the past days. So basically, I just will spend one slide in introducing my unit, which is within the Department of Cardiovascular Sciences and Public Health. Basically, my unit is serving biostatistics and medical statistics within the medical school. It is organized in three labs, and The three labs are, one is focusing mostly on clinical predictive model and is using also omics data. The second one is mostly run a mathematician and working on developing new models and methods in artificial intelligence, in particular language processing. The last one is the one which is really focusing on omics data analysis and is called statistics in experimental science. Most of us are biostatisticians, so we are really focusing on developing new models, starting from biostatistics, and also to apply those models in real world scenario. We have a medical residency program in medical statistics for medical doctors and a PhD and 2 master programs related to this field. So we just briefly go to illustrate what are our methodological issues that we are trying to deal. The first one is about general set of clinical predictive modeling. Of course, when you do prediction in a clinical setting, you have really some data, sometimes not that big data. Then you have to define a function that crunch those data somehow, and with the final idea to predict, let's say, an outcome. The real goal, the real issue for us is to have those predictive models as possible, because those models are then used in the clinical practice to stratify the risk of patient. And sometimes, and we have seen that in the COVID epidemic in Italy, at the very beginning, they really can have an impact on the decisions of clinicians in patients. Of course, when things are getting a little bit more complicated and we put omic data into the picture, you have the problem of dimensionality reduction, and that means try to avoid overfitting, feature selection, and also the issue about the different models that you can use and how to decide which is the best one. Or maybe the other idea is how to put all those models together, ensembling them in one single model. So let's go just a little bit deeper in this, and we have, as I said, we have data from very different sources. Sometimes those sources are even not structured, like we work straight from the discharge letters from the PDF. Sometimes are organized, but anyway, all of those data are really more or less overlapping information. Some proxies that we use in making prediction are just proxies of some things. I had a discussion yesterday about the concept and the use of race and ethnicity in predictive models, and they are just a proxy, of course, of some genetic, but also some social and environmental factors. All of them are, so the point is how to validate those models, how to extract and consider all those information, and how to evaluate attribution and each different set of information gives. Is really improving, or is just doing things like as before. The second is how to put together a model, how to choose them. I said we are using a lot of information from different models, like for support vector machine, neural network, random forest, and like a really a huge number. And then we ensemble them, and we weight the contribution that each single model gives to the to predict the outcome by doing a robust feature selection. And in this sense, the idea of model weighting is really promising, and we're really working on that. The last set of models that we are trying to use is to incorporate machine learning to construct the best probability score functions. What is a propensity score function? Is the idea to use observational data to from different coming again from different sources in a way that you isolate the confounding effects of all factors. So suppose I'm interested in evaluating the effect of genetic information on the outcome alone, variables with all the other information, then I have to construct this kind of propensity score. And again, superlearner and assembly model are really, I would say, the future for propensity score construction. In clinical setting, this means that we call this as post-randomization, because at the very end, everything is like can be treated as if the data would be coming from a randomized trial. So clinical perspective, this kind of models are really very well interpreted and again very of interest for the clinician. Just a few slides about what is going on, in particular in the statistics for the experimental science lab. It is really a growing sector in my unit. The idea is to exploit the potential of deep learning in omics data, getting very, very well refined predictions. And we are working on several projects. I'm just here. We have the project with the neuroscience department about muscular dystrophy. And this is a very exciting project because beside the richness of the information, this is perhaps one of the largest database in the world with 800 patients of this rare disease. Then we are working in the multiomics integration, and again using in particular Bayesian modeling and Bayesian machine learning. This is very applied in this field because there is already information available. So we want to exploit that information to identify the molecular markers which are mostly impacting the biological processing. Then with our colleagues from the anatomopathology, we are working on, again, in a fairly population of pleural mesothelioma. And again, here we are, the goal of this project is how to integrate histochemistry, radiomics, genomics into a single model to understand immune checkpoint inhibitors effect. The last one is about cryomics. Here we will use causal machine learning approaches, and this is about the exposure in vitro and in vivo to different temperature, and then rewarning with the final goal to features detection and biomarker discovery. Okay, this is it, and I would be very happy to interact with you later in the discussion.
Thank you very much, Professor Gregori. As a matter of fact, I will write you an email because I have looking. You just gave me an idea for a possible research project, but I will email to you at the meeting. Thank you very much. And I think the field of integrating, the idea of integrating different sources of data, which is not just genomics and transcriptomics, but the clinical and all the imaging and whatever other source of data is crucial to develop predictive models for specific both monogenic and polygenic or multifactorial disorders, or for cancer. At this point, sorry, it's my pleasure to give the word to Professor Julie Hussein, who is assistant professor at the Department of Medicine at the University of Montreal, which I suppose is in Montreal as well. You should turn your mic on.
Keeps happening to me. Thank you very much. Can you hear me now?
Yes.
Yes. Okay, good. Okay, thank you very much. Yes, I join my colleagues in thanking everyone for this, for the organization of this workshop, and also for the invitation to present here. So I am going to start with telling you a bit about my background. I don't have a slide for this, but so I was trained in population genetics and bioinformatics. So mainly during my training, my world revolved around statistical models to try to understand genetic diversity in different species, mainly humans, and my work has been applied a lot to health. So understanding how, for example, natural selection would shape the genomes and potentially lead to adaptations, but also to misadaptation to the modern world. So during my training, there was a lot of also of statistical genetics, and up to my postdoc, I came into realization that all of these statistical models were great. We have learned a lot from them, but they're simplification of the world for sure. And it came to a point where I felt like, for example, just the simple thing about biology of, interacting proteins and how this would impact the phenotypes, where all the models in statistical genetics would take, for example, one variant at a time and measure their effect on phenotype, was really a limitation. And of course, the fact of adding interaction in those models would lead to burden in terms of computational power and so on. So I saw a big limitation there. And at the same time, I learned about all these new models of machine learning, more data-driven as well models. And I saw a very high potential for this. And so starting my research group 4 years ago at University of Montreal, I decided to explore, along with collaborators at Mila in Montreal, possibilities for translating these for using machine learning and deep learning that would allow for interaction effects naturally into the research of statistical genetics or the team that we went. However, what I realized very, very quickly is that although the idea is great, there's a lot of challenges to do machine learning for prediction in genomics at the individual level. And so sorry about, I decided here to talk about these challenges because I thought it was interesting for this panel. So sometimes it's better to have a talk with lots of results, but like for today, I decided to focus more on these challenges and our way through them. We haven't solved them all, of course, but we've been thinking a lot about them. And here I'm showing the picture of two of my students that are working hard on these right now. So the first one is the fact that we don't really have big data in health science in general. We have fat data, which means that we have a lot of variables. And the talk just before mentioned, lots of different data, types of data, modalities, and so on. And that adds to that, fatness of data. Whereas we have, in general, small sample sizes, which means that we have less patients than features that we're measuring, especially in the multi-omic era we are in. Then we have data sets that are imperfect. Sometimes when we talk with machine learning experts, they see the data as a kind of a simulated data set where everything is complete. However, we know that those technologies, despite being great, they are doing mistakes. So there's noise in the data, and there's also missing data. The data is sparse, and so on. we have this issue where some machine learning algorithms work very well, but we have no idea why. And basically, if you look into and if you try hard to understand what's going on, most of the time the interpretations are not in line with what you would have expected if you would have done a kind of statistical model where you control everything and so on. Then all of a sudden the explanation is somewhere else, and often in biases of your data. So that's very concerning. And finally, there's this idea of trying to integrate a lot of different data types. As mentioned, omics data, but also clinical data, sometimes imaging and so on. So these are the challenges that we're facing. And so I don't want to be to look like I'm saying that there's nothing that has been a success in deep learning and genomics. There's a lot that has been successful working with convolutional neural networks and recurrent neural networks to understand sequence variability. But all these models, they look at the genome and the genomic data as big data because they are trying to learn on a large genome, lots of examples of motifs in the genome and so on. So we're doing base calling like this. We're studying mechanism of transcription, of methylation, promoters. But all of these are, elements of the genomes that are trying to be predicted and not outcome for a specific patient. If we are trying to predict on a patient level, then this becomes very more complicated because what we have as the features now are the number, for example, of genomic loci, SNPs, and then we have fewer participants than we have loci in the genome. So this is why it's a problem, and the problem is the fact that then we need a lot of parameters to fit for our models, and some tricks have been found in deep learning, for example, where, for example, in an image you could use those filters that go through the images where the parameters are the same, because locally having an eye, regardless of where it is in the picture, it's still an eye. But the problem is in the genome of the one individual, you don't have this stationarity property. So if, for example, you have 3 SNPs next to each other or in your data and they're AAT, well, this AAT doesn't mean the same thing if they're on another chromosome in another gene. So you lose this ability with genomic data to have these models working well because of this lack of stationarity. So you finish with, for example, if you're trying to predict with a neural network something. So here you have, for example, individuals, their SNPs, or any features that you want to include. Then you have this first layer, and this is an explosion of parameter to link it to your model that will try to be learning, for example, the risk of this individual. So here, imagine that we have the genotypes data in these features. We have a lot more features than participant. So with my colleague Yoshua Bengio at Mila, he suggested one solution that was really nice, that I felt was really interesting, was that let's not fit these parameters of the fat layer, but let's predict them. Let's predict these fat layers parameters, and let's not predict them just from somewhere else. Let's predict them from the data, from the data as a big data. So now what we are doing is that we're taking all these features. Now they are the examples, if you want. And we have some kind of representation of each of these features. So we have a lot of things that we know about SNPs, for example. And so that we can use as an embedding. And then we use this embedding in order to predict those parameters of the fat layer. And if you have an autoencoder, for example, and a decoder, you have a fat layer as an output as well, and you can do the same. You can do it jointly and so on. So in principle, it's a nice idea. And this feature embedding is really at the key, is really at the heart of this new way of thinking. This is a kind of an auxiliary network that we're using to predict these fat layers parameters. It can be pre-computed thanks to expert knowledge that we have in the field. It could be learned offline, on another task. For example, trying to predict something about the SNPs. And it also can be learned jointly with the rest of the architecture that you want. We tried this. So at first, what I thought was, let's try this on a very simple task. If you look at genetic data, well, it will scream ethnicity to you. So we used the 1,000 Genome Project data. So there's genetic data from 26 population worldwide. And we thought, okay, let's see, this is a fat data task because there's a lot of SNPs here. And we have only 3,450 individuals. So let's try to see if it can work. And it did work. So it did work quite well. We were expecting a very high accuracy on this task from what we know about population genetics. The mistakes made sense. It would confuse potentially individuals from Great Britain with individuals coming from France, and so that would make a lot of sense. And my student re-implemented it as well, and you have the implementation here in PyTorch. And Camille, she also verified generalization capability of that approach. So she took other data sets from genomics that were done at different times. So this is a older data set with like less data. This is Carthagène data set, is a biobank from Quebec. Here you have an excess of individuals from the French-Canadian population. So we wanted to see if that is not in the 1,000 genome. So we wanted to see what would happen. And basically during these experiments, it showed very good generalization capability. However, we discovered a problem that we knew about, but it went in clear that we had this, that they would cause, missing data would cause some very strange effect. For example, that French Canadian would be classified as Mexican if they had too many missing data. So here, it's a toy example. Clearly, we're more interested in health-related issues, but here we can really see it, like French Canadian are not Mexican. And so what's going on? During that time, when we discovered this, we saw that the model was very, very sensitive to missing values at a certain point of missing values. And so we included in our approach kind of an input dropout way of training. So more classically, we would, in population genetics and statistical genetics, we would impute the data. We try to get to have our model being robust to missing data, so being able to train, to train with missing data and to be able to give right answers. And what we could see is, of course, if you remove too much, you don't learn as well. But if you remove not too much, but and you have a lot of missing data, sometimes you can even do better than the models that have been trained with complete data with a lot of missing data. The last thing, another last thing that we did with this implementation of the diet network with population genetics is that we were trying to interpret the results. So in population genetics, we know that, for example, taking common variant and looking at their frequency is something that tells us a lot about differentiation between populations. And we wanted to see if that's the same that our network did see. So for each SNP, for example, we can have an attribution. We decided to take the integrated gradient, which is an approach that has been published and used a lot with deep learning. And so we have a score indicating how useful a feature is to make a prediction. And to our surprise, here you see the genotype frequency. So these are the kind of common SNP. And here you have the rare SNPs. And it seems like our model is basing their decision on rare variation, whereas in the kind of more classical models, we are really basing this on common heterozygotes, which was surprising. So we thought, okay, maybe this is not right. Maybe this is just noise and there's nothing, that's not what it's really learning, right? Because we are computing these scores, but we were not sure. However, we showed that it's actually true. So here you have the situation when you remove at random SNPs in your model. And here you have the situation when you remove them based on attribution. So the highest attribution you remove, and we see very clearly that we are losing our predictive power. So clearly this was right? We were learning, the network was learning something based on rare SNPs, which is different than what we have been developing from before. But it's very interesting because in terms of phenotypes, we know that rare SNPs may have a very big impact. And with the current models, we are really going short with the rarity of SNPs. So we are now in the situation where we are trying to do the same with phenotypes. And here we chose BMI because we wanted to merge this with other type of data. For example, we had seen this research where they showed that the built-in environment would be useful, and so to predict BMI. And we thought, okay, but what if we could join built-in environment and also genetics? To be honest, we started to try only the genetics just to see first if we can compare ourselves with polygenic risk scores, for example. And we are not that bad. I mean, it's very similar. We're clearly still working on the model. Here we're comparing it. Also, it's not the fairest comparison because the polygenic risk score that we used a test and train set, but we don't know the train test from the publication. So we kind of have to assume, and here we probably have leaking for the score. If we do exactly the same, so on all participants, we have leaking in both models, we do better. But so this means we are overfitting, they are overfitting. Still, a lot of work needs to be done, and so on. So I'm going to skip this because I see that I talked for a long time, but I added this slide at the end when I was preparing this presentation and thought I would skip, but actually I'm not skipping it because I saw from the previous presentation that this might be one of the main interests, and it's a very important point that we are also trying to develop. So in the idea of genetic risk scores and so on in that field, there's this issue where ethnicity, and you touched on this a bit, where ethnicity is a very important component, and where the scores that are developed in European populations where we have the most data is not as good as it's not performing as good in other populations. And so this whole thing that I showed you about trying to predict population is not just a toy example for us, it's also a way of potentially being able to integrate this aspect of ethnicity. So let's say we want to predict a complex phenotype. Well, we would like to use a domain adversarial neural network to make sure that we do not predict ethnicity, and that anything that is predictive of the complex phenotype is not necessarily predictive of ethnicity. So with these domain adversarial neural network, we feel that this is a very good way of controlling for population structure, for example, in this case, but also to control for sex or gender, recruitment centers, or technologies, as was mentioned before, technical batches that we have, this whole thing about imperfect data, but also time, seasons, depending on your task, whatever confounding can be included here. So this is it. Thank you very much for your attention.
Thank you very much. I have a comment on the critical the criticality of rapes. Actually, we have this game that when we look at West samples, we try to guess the ethnicity. And if you look at the rare SNPs, so the ones that you pretty much guess it correctly, because if you find 3 or 4 variants that are 1 in 1,000 in, let's say, Africans, and 1 in 100,000 or 0 in European, you're pretty sure that the last name will be, or the birthplace of the parents will be Nigeria, and you end up picking it up. I have another comment, but I will make it specific to you afterwards.
Okay.
Thank you very much. No, because we said this discussion afterwards. At this point, it's the turn of Professor Sperduti, also from the University of Padua. He's professor of computer science in the Department of Mathematics. So we have all the departments, the School of Medicine, the mathematics, engineering, and whatever, just to stress the multidisciplinarity of this roundtable. Please.
Thank you for invitation, for the organization of this very interesting roundtable. So I will not use any presentation. I will try to give just an idea from a very high level of what has been my involvement in this kind of problem. So my background is in computer science, and since the beginning of 90s, I was doing my PhD in neural networks, because at that time neural networks was very popular. And since I was a computer scientist, I was focusing on how to use neural networks, not just on vectors, but also on sequences, trees, graphs. So from the very beginning, I was interested in extending the capability of neural networks to structured data. And then, as you may know, history was not so soft with neural networks, in the sense that there was a period when neural networks were considered too powerful, and there was not enough data to train in a proper way a neural network. So kernel methods were actually dominating the scene. So, of course, I was interested in neural networks, but I have to also cope with the wave of the kernels wave. And so, guess what? I was again studying kernels for structured data. So, given this background, I was asked to contribute to a strategic project of the University of Padua on personalized medicine. So, so the idea was to try to, of course, as some of you already have stated, to use genomics in order to explain why some diseases are there. So what is the correlation between genes and disease? So my task was to try to understand how basically kernel methods or machine learning approach could help in this kind of large goal. And so we, with my research group, we focused on prioritizing disease genes. So the idea is that, okay, you know some genes that is positively correlated with some disease, and then you want to discover if there is any other gene that is actually involved in causing the disease. So basically the guilt by association principle or hypothesis was at the basis of our work. And kernel methods were actually very useful in this kind of task. Why? Because in this way we could leverage many protein-to-protein interaction information. And so just to make an example, in the first system that we developed that's called SCUBA, we use the Human Protein Reference Database, the BioGPS, pathways, and string. And so the idea was to focus, of course, on the relationship among genes. And so each database was encoded as a graph. So this could also be done for all general database where you have information about genes. And then a kernel based on graphs was actually a kernel that was measuring. So what is a kernel? Sorry, maybe I'm a bit too technical about that, but basically a kernel can be understood as a similarity or dissimilarity measure among two items, two entities. And so if you consider a node as an entity, a kernel is actually giving you basically a similarity measure between two nodes in the graph. And this can be done using the topology of the graph. And so here we are back to the guilty by association hypothesis. So using different databases, so different graphs, we were actually trying to leverage the information from different databases in order to find similarity between genes. And then having a short list of genes that are considered to be guilt, in the sense positive correlated with the disease, the task was to try to rank the remaining genes in such a way to see if it was possible to identify additional genes that were actually positively correlated with the disease. So in order to do that, we transformed the databases that we were referring before into graphs. Then we applied this kernel approach to these graphs. And then we were combining all these kernels using a technique that actually allows to combine in an optimal way the contribution of each graph. So this was done on several data sets, and the system was actually performing quite well. Especially, we tested an unbiased setting, experimental setting, where what we did, we were actually considering we considered all versions of the databases that were reporting the correlation between genes and disease, and then we stopped the training on a given date, and then we used the information that was discovered after that date as test data. And so our system at that time was talking about 2018, it was performing quite well. And another feature of our system was the fact that we used some assumptions and some mathematical, let's say, solutions that allowed the system to scale quite well with the number of kernels, so with different similarity measures and with the number of training examples. So the system was actually performing quite well. However, there was a problem in the sense that each database was considered singularly, and then only a posteriori it was merged with all the other databases, and all the kernels that we were considering, they were actually kind of eliciting global information about the graph. While it would be better to use also some local information in order to avoid some artifact that can come up from having, for example, one gene that is correlated with many other genes. So in this way, is going to dominate the learning and also the outcome of the results. So what we did was to devise a new system called DIGI. DIGI is actually merging all the graphs of the database all together and considering some association as important and some other as like preferential, so soft connections. I skip all the technical details even because I do not have time here to go in deep to that. And basically, the main difference of DIGI with respect to Scuba is that now all the databases are combined all together in a huge network. And then we developed a new kernel that was actually using local features of the neighbors of each gene, which means looking at the information and the different databases on strictly related genes. And this was actually giving a further boost to the performance. I would say that this kind of approach was quite effective. However, if we consider some of the issues that have been raised already by the colleagues, like interpretability, you can sure try to interpret the result and understand why a specific ranking is given in output and not another one can be done, but it is very hard to do that. Another issue that is very important is that I think also, I mean, I'm sure that in general, even convolutional neural networks for graphs, that was one of the techniques that was mentioning, they have the problem that they basically elicitate global information. The way they work, this is actually the case. And also they tend to use compact information in the sense that they are not able in general to find disjoint causal subgraphs that can be the origin of the effect you are interested in. So I think there is a lot of work to do also from a methodological point of view in order to find not just a cause that is kind of elicited in a global way, but several causes that can jointly find, explain a specific effect. And on top of that, there is the difficulty that is given by what Julie was mentioning, that very often data is not so large. And so there is also this kind of problem that needs to be addressed. I think that we are in the need of merging bottom-up approaches that are based on data with knowledge that is top-down. And only in this way we can really try to develop models that can also be explainable. Because otherwise, if you do not, by construction, give some interpretation to what the model is doing or what the components of the model are performing, it will be very difficult to find explanations. So this is it, I guess.
Sorry. Thank you very much, Professor Sperduti. And I admit that I had trouble in following all your steps because due to my ignorance in the field. However, I think that there are a few aspects that I will try and to address with specific question afterwards. And it's very interesting, this kind of approach, which is, especially for polygenic traits at this point are the main one of the most important fields of genetics and genomics for the rest of the century. At this point, I have to say, last but not least, Professor Jérôme Waldispoel. Have I pronounced it correctly?
Yeah, that's correct. Yeah, I mean, there's so many ways of saying it that anyway, it's fine.
My French is a little bit rusty. I can survive in French, but certainly my accent is not adequate. Anyway, and he's our last speaker for today, but for the session. He's associate professor in the Department of Computer Science at McGill University in Montreal. And please, go ahead, you can see. Not my point.
So you should see my slide now.
Yes, please.
Perfect.
Yeah, perfect.
So thanks a lot for the invitation. I'm very glad to speak here. And initially, I'm coming from Nice, so I'm perhaps historically closer to Italy that my name might let you think. So I will speak today about the work I've been conducted in the last decade about try to improve using algorithm and machine learning the analysis and prediction of RNA 3D structures. But why RNA first? It's because RNA, beyond the messenger RNA function where you carry a gene that carries information and bring enable to copy DNA and to be translated after into proteins. RNA are performing tons of function, and we're just discovering more and more these functions we uncover a lot of this function in the last decades. Of course, protein synthesis, splicing, but also in gene expression and guiding mutation in genomes and methylation. And often the key behind is that structure for this non-conic function is often key to understand this function. And as such, my work has been trying to leverage both technological and biological ideas to make a better use of the biological modeling techniques we have and algorithm and computational techniques that we can use to analyze these structures. And the key idea beyond my research here is really to try to represent RNA structure as a graph, importantly including what we call a non-canonical interaction with very sophisticated and detailed view of the structure, but it's still a graphical model, and use it to accelerate the prediction of the 3D structures, but also the binding of small molecules using, in particular, machine learning technology these days. So the long-term objective of this, of course, is to help, as I said, the understanding how these molecules are formed and how they interact with other molecules. And so the first steps of the research program I'm conducting here is really to first, starting from a sequence, how can I predict such a 3D structures, and then look at how small molecule will eventually bind this structure to modify its structure. So the modification can be at 3D levels. It can be more dramatic, like changing what we call the secondary structures. But what is important is understanding how this molecule will bind the RNA and change its function, and eventually try to use this small molecule to better control the function that is carried by the RNA. But why do we care that much for RNA and targeting RNA as a drug target? Well, if you think about it, most of the drugs that are available these days are targeting proteins, but proteins represent only 1.5% of the genomes, which is a tiny fraction of what RNA represent. We estimate that up to 70% of the RNAs of our genomes encode for RNAs. So we don't know exactly each function of them. Some may be more useful than others, but that gives you still an understanding about the potential for druggability of the genomes that is opened by targeting RNA rather than proteins. And this is thought really to open a new era in the pharmaceutical industry. So now to come back to RNA structure, how do we, what are we doing, and how we do, what are the techniques that we're using to achieve the agenda I just presented? Well, something which you have to understand with RNAs, like they're highly structured and they have a hierarchical organization of the structure. And importantly, if you look at what we call the secondary structure, that is the ensemble of Watson-Crick and Wubble base pair finding structures, all RNAs have a highly conserved secondary structure that can be represented as a planar graph that is highlighted here on the right, that is used during the folding of RNA as a scaffold for the 3D structure. And the property of being a planar graph for these RNA secondary structures enable us to apply very efficient techniques in computer science, namely dynamic programming, to quickly compute the scaffold, and then after that, use it to predict the 3D structures and doing molecular and biochemistry or structural biology analysis. So typically, the classical approach to analyze an RNA, and that's is still the one using nowadays and still developed, is the following. So first, you start with the sequence. From that, you predict a graph that represents the secondary structures. That is, after that, used to predict the 3D structures. And once you have the 3D structures, we can start to predict interaction with small molecules using docking software. So what you have to understand is that if the first step of going from the sequence to the secondary structure is relatively fast, it's a cubic time algorithm that runs in seconds depending on the size of the RNA we're looking at. The step of building the 3D structures is much more computationally intensive, and the accuracy is not there yet. Even when you have it, then after scanning full libraries of molecules and try to see how they can bind this RNA, control these expressions, is very, very expensive. And if binding one single molecule can be fast, binding thousands, tens of thousands, millions of molecules is much more expensive. So the approach that we're leveraging in the lab is to merge together two ideas. The first one is like going beyond the 2D structures and looking at what I was mentioning earlier, this non-canonical base pair. And once we have this graph that is an expanded graph, so more complete graph of the different type of interaction occurring within RNA, we can then leverage the machine learning and in general AI technologies to make a better use of this information. So a couple of examples. So basically what I'm saying this is like, when usually we think about the base pairing, we think about only the Watson-Crick and the Wubble, but there are actually up to 12 different types of interactions that can occur. And we can also enrich, again, this vocabulary. So typically, this RNA enables us to re-expand the annotation and the interaction occurring within RNA. As you see here, you have this classical secondary structures, and all the colored edges are the non-canonical ones. And usually what's interesting is that these non-canonical interactions are located in secondary structure elements that are basically very highly structured blocks of the RNA that are used to shape the 3D structure of your RNA. So trying to understand how these non-canonical interactions are located in structures, isolating them as a motif is potentially the key to better predict the RNA 3D structures. So what we've been trying to do in the lab was trying to being able to better predict these 3D motifs, to analyze and make a catalog of these 3D motifs first, and then afterward being able to predict them much more efficiently. And what we see indeed is like, when you look at the base pairing network that we highlighted in the herpes of the junctions, there is a strong 3D conservation of this network. So the first approach we're doing was applying here a graph neural network to be able to help us to identify this motif that were highly conserved at the graph level, but ultimately we were hoping at that time conserve also the 3D structures. And the idea was is relatively simple. It's basically using a graph neural net to embed each RNA graphs into a multidimensional space, and then after classical clustering techniques to identify the clusters. And what we've been able to find is like building this class of motifs where the motifs were similar but not necessarily identical. And when we looked at the conservation of the 3D structures, we indeed saw that the 3D structure was conserved. So that gives us a good database of motifs we can use after that to do predictions. So when you have this database of motifs, we can design techniques to make better prediction of this motif. And typically, the system we've been put in place was to start with a single RNA sequence, fold it using classical secondary structure prediction algorithm, see where are the potential insertion sites for the different motifs, and score them using a Bayesian network. And once you have this, that enables to really boost drastically the prediction of this motif within the RNA. But that's not it. So once you know where the motif could be, you need to have a coherent prediction. And this is where we build this RNA MYP framework that consists in taking multiple secondary structure, possible secondary structure for your RNA, look at the potential insertion of the motif you can do, and bring them together and try to find the best fit between the best secondary structure you can find there and the motif you can find here. And ultimately, when you build what we call here a 2.5D secondary structures, when you have the Watson Crick and Wobble here, the stems and the motif in between, we can use it to make accurate 3D reconstruction of the RNA. And then the last work that we've been conducting was to say that, well, now that we know where are these motifs and we know how to predict the structures, how can we leverage this information to predict the binding of small molecule? And that's what we've been doing in this software called RNA-Amigos that also leverage the same idea of using graph neural net, where the idea was to encode a motif or RNA graph using a graph neural network. And then after applying a simple neural network, a multi-layer perception here, to try to maximize the probability of generating what we call a fingerprint, but if you want a molecule that fits the motif we're interested in. So overall, the pipeline we've been setting up is really developing tools to accelerate all these drug discovery on RNA targets, where first we analyze 3D structures and build database of motifs. We use this motif and use this database of motifs to make better prediction of motifs inside secondary structures, and then assemble them into a 2.5D structure that enable us to compute 3D models. And when we have these 3D models, we can also use this RNA-Amigos software to predict where it can bind. So there's multiple collaborators there, but just beyond closing that talk, there's another project I just want to quickly discuss in 30 seconds, because I think it goes beyond what we're doing this, but shows new area. It's not a project that was actually also funded by Genome Quebec and Genome Canada through the GAP program, and that shows the potential of partnership that can be built in machine learning and biology and citizen science. So the idea was to integrate inside popular video games some puzzle that enable us to make a better analysis of biological data. And ultimately, the idea is to embed a small puzzle that enable us to ask people to help us to align, in that case, some rRNA sequence alignment task, and use it to analyze and better classify RNA sequence from microbiome projects. And the project is highly successful, enable us to engage more than 2 million participants and collect solution for this. And why are we doing this? Is because ultimately that's a way to collect more manually created annotation that can be leveraged after that by machine learning techniques to design better algorithm and improve the accuracy of current techniques. So that's it for me.
Thank you very much for your intervention, which is very interesting. And also, I will have some questions for you on specific research issues, because we study mitochondrial and in mitochondrial disorders, mutations in the tRNA genes are quite frequent, which is something that's unheard of in Mendelian genes. And but we can talk about this later. So at this point, we have gone through the first round of presentation. If I have understood correctly, there should be a small pause now. Please correct me if I'm wrong. And after the break, we can start with the specific round of questions for each speaker, right? Cioè, lo dico in italiano, abbiamo fatto più del primo giro di domande, di presentazioni, e se ho ben capito ci dovrebbe essere il break adesso e poi passiamo con...
Esatto.
Esatto, ok.
Yes, we can have maybe 10 minutes of break, and then we can at maybe 15 to.
Okay, so now it's 4:30. It's 4:30 p.m. We can meet at 4:45. Is it okay?
Perfect.
Okay.
A 4:45. That is maybe a 11:45. I think. Okay. See you later.
Easter time.
Thank you.
Okay. Thank you very much,
Grazie,
everybody.
professor.
See you in 15 minutes.
Professor Salviati, mi
Sì?
scusi, solo per dirle che io alle 17:30 oggi purtroppo devo staccare, ma ci sono sempre connessi i miei colleghi dell'ambito tecnico, quindi per qualsiasi cosa, ecco, vedo c'è Matteo.
No, no, vabbè, ma non credo che ci sia niente di particolare
No,
che
infatti, neanch'io,
pare
anche
che andando avanti abbastanza bene, insomma,
mi pare di sì, anzi.
cioè, le presentazioni sono state interessanti, adesso facciamo il giro di domande. L'unica cosa è che era una domanda più specifica per la Julie Hussein rispetto a quella che l'avevo
Le aveva inviato.
che le avevo inviato. Beh, insomma, vedo adesso gliela preannuncio perché tutto sommato ehm, eh,
Ma sì,
eh,
secondo me, voglio dire, se è sorto qualche spunto in più ascoltando le presentazioni, come è stato anche ieri.
ci sono un paio di cose generali che volevo fare alla fine. eh, ehm, sul data overfitting, che è una cosa che hanno menzionato qualcuno, ma e, e, eh,
Sì, penso che eventualmente chieda, ma non penso che ci saranno problemi.
sì, insomma, ecco, basta, poi noi finiamo a che ora? Verso le 6, insomma.
Sì, ma anche insomma a seconda di come va avanti la conversazione, se volete finire prima o dopo, non penso che ci siano problemi, ecco.
Vabbè, non vogliamo mettere a prova la pazienza dell'audience.
Certo.
Adesso, quando il collegamento adesso su YouTube è bloccato o sta ancora andando?
Questo penso che stia andando.
Quindi è meglio che non diciamo stupidaggini, porcherie, perché sennò ci
Rimane traccia.
rimane nella storia come raccontare figure da chiodi. Vabbè, grazie a te e vabbè, insomma,
Grazie intanto, comunque per qualsiasi cosa siamo qua.
no, vabbè, ma insomma,
Non per dirle questo.
speriamo che non ci sia bisogno.
No, sì, per qualsiasi evenienza tecnica, ecco, era solo per dirle
Vabbè.
questo.
Il professor Gerosa si connetterà in qualche
No,
momento?
ho avuto un problema oggi e proprio non ce la fa.
No, è tanto sapere perché se c'era da aspettare, convocare o fare qualcosa, se no noi andiamo avanti fino a che...
No, andate pure avanti. Oggi purtroppo non riesce proprio a connettersi.
Vabbè,
Grazie ancora intanto.
niente, grazie a voi perché tutto sommato è stata non inutile, che già ci sono un paio di robe che si
Quello è l'obiettivo, riuscire poi sicuramente ci faremo anche sentire noi con l'obiettivo magari di fare uno scambio di contatti o quant'altro, ecco.
Vabbè, grazie.
Grazie e buon proseguimento.
Vabbè, 2 minuti per cui adesso si connetteranno.
Esatto, infatti.
I am back.
Chi sei back?
Is back? Mauro Conti.
Mauro, ecco, perché avevo lo schermo semi... Io mi scuso della domanda generica, ma facevo fatica a fare una domanda più specifica, non avendo idea di fosse lo specifico argomento, e spero che non sia troppo stupida come domanda. Solo...
Intendi a me nello specifico?
No, perché gli altri qualcosina forse un po' meno cretino sono riuscito a farla.
No, in realtà ho trovato una risposta simpatica nel senso
non
che,
volevo,
ripeto,
perché a volte appunto queste cose che bisogna fare delle robe preordinate, a volte si dice di fare delle domande che sono in effetti alla Julie Hussein gli ho fatto una domanda che è cretina, infatti volevo chiedere se o comunque insomma è poco rilevante, ce n'era una più specifica che volevo fare.
Siamo comunque sempre su YouTube.
No, lo so che siamo in streaming, per cui commentavamo prima con Giorgia di non dire stupidaggini o commenti poco simpatici, perché siamo in mondovisione, siamo una figura da chiodi notevole. So, I think everybody, we're still waiting for Professor Conti to come back.
I'm here.
No, but I see. È vero to hear, but there's two of you in.
Yeah. Just I have two devices.
Okay. You are indeed here. No, I was seeing somebody with another profile showing up.
That is a clone, a genetic clone.
Exactly. One of the problems with genetic identifications that are chimeras, identical twins, and so on. But this is another topic for another roundtable. And okay, so I think it's a quarter to 5. We can start the first. At this point, I will have a first round of questions, and for Professor Conti, who is indeed here, and besides the issue of cybersecurity and before let me add one more comment. Please, if you have any comments and talking to the other speakers, feel free to speak up in the stage of the discussion. So if you have if any of you has a question for Mauro Conti, please feel free to just raise the hand or take you or ask, because I would like this to be as much as interactive as possible. Anyway, so my for Mauro Conti is that in one of the paradoxically one of the main issues besides security, which is a problem because we had I'm saying it for our Canadian, one of the two Padova hospitals had a major breach in the security a couple of weeks ago, and some data of some patient data were actually leaked, and I don't know how the whole thing ended, but so this is one major concern. But paradoxically, the other problem, one of the other problems we are dealing with is the fact that single whole genome analysis in a trio, so patient and the two parents, produces several hundred gigabases of data. So I would like for you, if you could comment on the issue also besides the security, also of storage itself of these huge amounts of data. Think that we run more than 2,000 tests almost 2,000 tests per year. If our IT people would go nuts if we had to do whole genome sequencing to all of them, and it's going to happen in a few years.
Okay, here I am. So yeah, thanks for the question, Leonardo. Actually, this is a very, of course, important observation. I mean, somehow, as Alessandro were mentioning in the evolution importance of neural network and data, of course, with the technological advances that we have now, it's not a huge problem. The storage itself, of course, I mean, if we want everybody's genome to be available and accessible for computation, that is a bit of a challenge, but it's not impossible to manage with the current technology. As you were mentioning, there are issues linked to the fact that these are very private data, and of course there can be a lot of aspects related to the security and the privacy of this data. So we store data in an encrypted way to grant the proper access control, the user authentication to access this data, and so on. But what I would like to mention here in terms of problem of storing a big amount of data, actually, you're aware for sure that DNA itself is considered as one of the possible way to store a big amount of data, because in terms of storages, I mean, if you want to store gigabytes, you need, I mean, at least few centimeters of devices. Okay, if you want just to measure in terms of weights, while a single gram of data of DNA can store much bigger amount of information. So to comment on this and to go on something which, for the disclaimer I made before, is a work I have done in collaboration with biologists, and here comes where some of the things I'm not fully aware of the biological part, but try to come up with a solution to not only store but also retrieve information stored on a biological material. And this actually, again, a work we have done with Waterford Institute of Technology, and where there are also people working in the biological sector. And the idea was to come up with a solution to store and retrieve information from biological data. And this was also featured by the MIT Technology Review. Again, please bear with me if I'm not fully sound and perfect on the biological description of the idea, but I would like to share this with you and actually get your comment whether you think this has been crazy. And idea, of course, this is a very kind of visionary, and we have to work a lot on this. And the idea here is to store information inside the plasmid of bacteria, and thanks to the conjugation, retrieve information and bring this information somewhere else where they can be read. And this can be done thanks to the mobile motile bacteria. So the storage should be done by encoding information to nucleotides. For example, we used, I mean, in this specific test we have done to the people that have taken care of the wet lab experiment, we use these Goldman encodings and then encoded the information that we'll describe a bit more later inside plasmids, which are stored, let's say, if we can, in some bacteria. And the data retrieval mode is basically as following. So some bacteria are here static with the information that we have stored in a whatever, let's call it a database, and then we send motile bacteria to move through this space where they, thanks to the conjugation, they can somehow retrieve, let's say, the data and move to somewhere else when this data is read. Just to give you, I mean, a bit more, I mean, information on this crazy idea. I mean, in keeping with the tradition, you probably know that we, I mean, crazy computer scientists, when we learn a new programming language, what we do the first program that we write is called as a hello world. So it's a simple program to give a simple output. So what we have done is we encoded actually the string or the information, if you like, hello world inside the plasmids of the motility bacteria that are stored or placed in this agar plate section here, and material are put here, and then of course they can move around, and they will reach a place C where the information can be read. And this is possible thanks to the conjugation with the bacteria that are inside this static agar plate. Instead of, I mean, showing this with beads, we have done some experiments. Actually, this experiment, this is not I didn't do, but people in the wet lab experiment have done. so we position on the 2 antibiotics. So actually see the details below here. So we use 2 strains of Escherichia coli, so Nova 2 and HP 101. I have no idea, I mean, what are the specifications of these 2, but they are basically 2 different bacteria that they are resistant to 2 different antibiotics. So what we have done is we put basically inside the agar plate, we put the Nova Blue, and this was surrounded by the streptomycin. So to make sure that Nova Blue will not go out of this square, let's say, and the motile bacterial will instead lost this because the motile is the other one, so the HB 101. So it will go through the, let's say, barrier of streptomycin, because that one, as you probably know better than me, is resistant to streptomycin. So we'll conjugate with the bacteria that are inside, we'll get information from the plasmid, keep moving and eventually go on the C area. On the C area, we have basically both antibiotics, so both the tetraclysine and streptomycin. So it means if a bacteria is present there, it means it has been able to be resistant to both antibiotics. So, and that is possible only if they actually in this area and they conjugate it with the other bacteria. So basically moving this from a let's say at an abstract level, you can really see this as a way of storing information in the plasmids of bacteria that are here, and you can have basically a way to read information from what you can call basically a storage area that can be, I mean, of course, moving from this, very pilot experiment to a real usable, let's say, storage system. There is still a lot of way, also because I don't remember exactly, but it takes a few hours, let's say, to make sure that these motile bacteria traverse a given specific area. And we came up with solution. Thanks to attractant and repellents, we can somehow, extract this bacteria or force these bacteria to move some specific paths, and they're basically building blocks that we can leverage to build storage system where DNA or general biological material can be used both to store data and to retrieve data. I cannot hear.
I was saying the problem, as the title of your paper says, the problem is the retrieval of data, because then
yes, the time.
So because basically you can't resequence the plasmid, because let's say if you're using 2 different words, hello and goodbye, once the bacteria have gone from A to C, then you can pick up individual bacteria, but then you have to resequence the data. So the cost of sequencing is...
Yeah, that is true. So what we have done in this, let's say, visual experiment, we put also some fluorescent material in the bacteria inside the, let's say, storage area. So if you see the fluorescent action here, it means they went through it and they retrieved the information. So somehow, if you like, still as a model, okay, just to simplify.
The model is
extremely, yeah, to extremely simplify, even visually, you would have just encoding 0, 1, and then just visually you can retrieve even without sequencing. But of course, that will be very, it's in terms of storage, because it means you retrieve only 0, 1. But if you are able to resequence here, then it means you have a storage location which is there, and you send some in the current memory or hard disk or any type of memory storage, you transfer data on what is called the bus on a computer. Basically, you send a command, you collect the data, you get the data back, and somehow we wanted to, abstract level to re-implement something with the biological material.
The model is fantastic. It's really nice. Very, very nice. Are there any comments, or shall we for Professor Conti, or shall we move to Professor Emad? And my question for him was basically what you showed us is a tool to integrate information and from not only genomics but also data and what about epigenetic signatures? What is the role? And I'm saying this because epigenetic signatures are still at the DNA level, which is much more simple to handle than the RNA level. So it's sort of they reflect the genetics reflects the transcriptome, at least in that specific cell type.
Yeah, it's a great question. So basically, the way that I like to answer this is that yes, epigenomic data modality has been used for finding either biomarkers of drug sensitivity or drug response prediction as a single data modality, mainly for some very specific drugs with very strong signal. But the way that it is usually used is in the kind of multiomics framework that in combination with other type of omic data. So in fact, there was a competition a few years ago. It was a dream challenge where they were trying to ask the community to develop different models to predict drug response. And they had provided many different type of omics data, including, for example, epigenomic, genomic, proteomic, transcriptomic. And after that, after they got the data and the results, they did a lot of evaluation to see what data modality as a single data modality, cell omic data, is most predictive of drug response. And what they found over all these different algorithms was that transcriptomic data was the one, and because it's an intermediate phenotype, right? So it's closer to the drug response, so it's more predictive of that. But then they also found out that if you combine that with other type of omic data, in some cases it can improve. But then the reason I say in some cases goes back to the very nice slide that Julie put in her presentation when she was talking about the challenges in type of this machine learning models. Because if you do multiomics data in a naive way, you are basically making your feature design matrix just fatter instead of making it taller. And this makes the overfitting situation in machine learning and generalizability even worse than instead of helping us. So another way that we, however, use the type of epigenomic data is when we're looking at, for example, transcriptional regulatory network that are involved in drug response, because there we can then combine it with other omic data more as a kind of a filtering step to get more evidence and more confidence in the relationship that we find, instead of just increasing the number of features and the number of parameters that then our machine learning model has to be trained.
Yeah, thanks. No, the reason I'm talking about this is because in the transition to clinical work of these applications, you need to take into account also the logistics of the whole operation. For example, in the past years, we were performing molecular analysis on RNA because it was simpler for some genes, but it was a nightmare to ship RNA from one center to the other, because you had to do it in dry ice, be very careful, and so on. So if it's possible to develop models which stick onto DNA, it's certainly much better. Am I pronouncing it correctly, Julie?
Yes, it's ça. Yes, good.
Is asking to
Yes.
to comment. Thank you. Please go.
Yes, I had a question for you. I mean, when you were presenting and discussing the fact that, cell lines, it's not perfect, but it's a control environment, and then you go to patients and tumors and real tumors. So how much of because the cell lines all have the same genetic background, right? When you do whereas the tumor, has the genetic background of the individual, but also has this whole heterogeneity caused by the cancer, like not necessarily causal to the cancer, but like derived from those defects. So how much do you think that this genomic background is impacting, this translation from cells to tumors?
Yeah, so this is a great point because in my opinion, the best that at this point we can hope for is to correct for this statistical differences between the data distribution that we're getting. But there are certain things that biologically are different between cancer cell lines and tumors. For example, you have the intra-tumor heterogeneity. Even if you don't look at, for example, the effect of tumor microenvironment is another factor that cancer cell lines can't capture. Cancer cell lines, as you mentioned, are homogeneous than cancer tumors. So even from the cancer cells that are present in a single tumor, they have, they're heterogeneous. They have not only at the genomic level, but at the transcriptomic level, proteomic level, all those things are different. So so I don't want to get into the single cell aspect of it, but if you just use at the bulk level, you get an average of all these different factors, and cancer cell lines don't have that, cannot capture that. So how can we deal with this? Well, better models of cancer. So that's one option. For example, patient xenografts, or at least solve this at the cancer model. It's expensive, but it's doable. But then the closer we get to the cancer patients, the expense, all those aspects and number of samples become difficult. Another way would be if we can disentangle this cancer tumors to kind of clean the signal and get it maybe more similar to cancer cell lines. Again, that opens up some doors, but it needs to be seen how well things perform. But you are right. It's as long as we have imperfect models of cancer for our training, somehow we need to account for that. So we can use, for example, ideas from transfer learning or options like few-shot learning or one-shot learning. But then again, if your test data, clinical data, is very sparse, and you want to keep it for evaluation, then that also becomes prohibitive. So all of these have like kind of pros and cons of what approach to take. But yeah.
Thank you. And well, Professor Sperduti, please.
Sorry, I can't hear you. I think you're still muted.
Yes, sorry. So my question is flowing, but more general. You were mentioning interpretability, explainability of this model. We know that with this kind of model, it's very hard to have an explanation. And currently, there are at least two different ways of approaching it. Try to use a good model and try to, in some way, using heat maps and so on, projecting the back some sensitivity to the output is possible in a way to give some explanation by highlighting some parts of the input as the major contribution to the output. Another approach is to devise models that by construction are prone to give an explanation. I think that in this area is very important to give this kind of explanation. So I wonder if you have any position or project for the future about that.
Yeah, this is a great point. So I just wanted to, for example, talk from this model here. So as you very nicely brought up, there are really two approaches, right? So you can design a machine learning model or a deep learning model that within the architecture of the model itself, you have integrated interpretability. For example, there are methods that if you have a deep learning model, you may add another layer that it's your pathway layer, and the connectivity of your edges are based on which gene belongs to that signaling pathway. So that's one approach that tries to change the architecture itself to make it more interpretable. Other approach that you mentioned is the one that we are taking there, where you first develop a model that does a good job on prediction, and then they try to perturb it to find out what feature, for example, was contributing to the. So in this particular project and some of the other ones, we took this the last approach to find what are the first finding a good model that does a good prediction, and then finding, which one was most important. We are also working on developing new models that can take this to the, for example, pathway level. So just instead of selecting input features, maybe how can we best select the top, for example, signaling pathways that are involved. But then we are also working on infusing interpretability within the architecture. Now, the problem there is that, again, domain knowledge in some way needs to be incorporated. So how can we make that systematic? One way would be to, for example, use a priori annotated gene-level interaction networks or signaling pathway involvement into the model. We are working on that, but the problem is if the network and if the pathway that we are having annotated and we know they're very incomplete and we know that they are noisy, they may actually hurt us than helping instead of helping us. In fact, a lot of approaches that are already existing in this domain that try to do that, the way that they approach it is that, they show that adding those type of pathway information does not reduce the performance of the model too much. So it doesn't help to improve things, it just doesn't hurt as much. So to me, that's still something that needs to be improved, because ideally, if we want to have architectures that within them there is interpretability, it needs to do better than baseline, not do too much worse than the baseline. So I think there is a lot of work to be done on that front, and some of it simply may be due to incompleteness of the a priori annotated networks and pathways that we currently have.
So just a short comment question. But then wouldn't this be also the opportunity to revise the knowledge? Because you have data that is coming from the ground. You are also designing your architecture such a way that you have a specific information that is explicitly coded based on your knowledge, not your prior knowledge. But then why don't take the opportunity to guess if you can revise the knowledge that you have, maybe using the black model that is performing better? Do you think this could be a viable way?
So if I understand correctly, I think your suggestion is that, let's say we take for example, pathway data set, bring it into the architecture and modify it, for example, remove some genes or add some genes in a way that it actually performs better, and that could be kind of a corrected version of the original pathway that has been done. I think this could be an interesting approach. For it to be convincing, however, it needs to be done in many different applications. So for one, to say that it is robust, it needs to be confirmed experimentally, in my opinion. Otherwise, still it's going to be. And the other variable that is hard to control for is to understand whether this better performance that we observe is truly because we have modified the pathway or the network properly, or whether the other components of our architecture is doing something with them.
Well, my suggestion was to use the network that is not using the pathways, explicit pathway information, that is performing in a better way as teacher to the other one.
Yeah, so I think that could be an interesting approach to augment some of the already incomplete data sets that we have. I guess. Nice.
Okay, thanks.
Thank you very much. And at this point, I have a question for Gregory, and And I like his presentation because it gave me it was really food of thought for many of the things we deal with daily, also in the clinic. And one of the problems with rare disease, we were discussing this yesterday in the gene therapy session, was the fact that sometimes it's very difficult to make outcome prediction based on the genotype. For example, there are some diseases in which you can't really predict, actually correct faithfully what will be the likely outcome of that patient. So if he will develop, he will have a fast progression of disease or a slow progression of disease, looking at the specific mutation causing that disorder. But so there is a clear genotype correlation. But for some other diseases, this is not the case. So you know that is the mutation that is pathogenic, but you cannot really predict whether he will have a severe progression or a mild disease, and this is crucial. And I like what you were discussing here before about your prediction and your tools to make prediction. But what are the issues on applying this to rare disease? Because for common disorders, you can have a large set of data to work with. But what about rare disease?
Yes, I think exactly that there are some really methodological issues behind the outcome prediction model in rare diseases, but in general in situation where you have a small set data set. I want to share some slides I prepared to help me going through these points, or at least to set some points. I'm wondering if,
yes,
yeah, wonderful. Okay, this is something, just machine learning and rare disease. This was a review published 2 years ago, and what I just highlight that both Italy and Canada are among the biggest producer of knowledge in machine learning and rare disease in the world, far beyond the US, but still on a very close level to China. What was wondering me is that the pattern of the usage of machine learning in outcome prediction in disease is not homogeneous. In some, for some diseases like cardiac diseases, gastroenterology, and hepatic, and so on, there are more machine learning techniques in studying rare diseases than in the general class of pathology. In others, like skin diseases, chronic disorder, and others, we are far beyond. And this means that basically there is some pathway related to the way that machine learning is around the clinics. So what are the issues? I would just focus on my on methodological issues, and leaving maybe the discussion about the biology to the floor. So I would say there are three in my view. What we call the horizontal data metrics, and unbalancing data and model validation in causal interpretation. So I think Julia already touched this point. This is very simple. We have more feature, more variables than observations. In particular, when we go clinic, we have, as it is a rare disease, we don't have too many patients. And when you put genomics into the picture, the number of features goes even larger. What is the problem here is that it's based on overfitting and variable selection and unstable predictions. That means it's basically one of the worst scenario. And the problem is also that you are at risk of losing interpretability, because techniques that are used commonly to overcome this problem, like I'm saying random forest is one of the most popular, are not really allowing a very good interpretation. So we will be focusing here mostly on regularization. I think regularization techniques are really maintaining, or the capability of maintaining the interpretability, because we are dealing basically, this is a very simple elastic net models. That means a regularized model where you have a regression. That means something that you more or less you learn in the medical school here in the medical statistics course, but you have some restriction of L1 and L2 norm on the value of the coefficient. This means that you basically shrink down the coefficient towards 0 for the unimportant features, and more important features are coming out, and you can observe them. But interpretation is still the same because it's given by the normal, I would say, coefficient. And these techniques are really very, very flexible. You can use normal techniques to validate the model. And this is an example about flow cytometry data. There was a model basically skipping the cell gating and substituting the cell gating with a predictive modeling algorithm based on ElasticNet. And you see this model outperforms with compared to other very simple regression that was expected and other models. So this also in particular for rare disease, there are some complex approaches, all of them based on modeling, and regularization techniques are a building block at the beginning for constructing the model. So the second issue was data imbalance. What does it mean? It means that you have suppose you have two classes and you want to set up a classifier, and you have a very small number of observations for one class and a huge number, overwhelming huge number of information, that means observation for the other class. At that point, naive classifier, they tend to classify the lesser represented class as wrongly, because in that way you minimize the prediction error. And how do you overcome this? You basically, this situation, you have a small number of observation in one class and all the others are in the other class. So you basically oversample, represent the smaller class, and you do that by resampling. This is random forest techniques is one of the most commonly used in this approach, because it's intrinsically based on resampling. And the goal is that you reduce the loss of accuracy in prediction due to imbalance. This means that imbalance is going high and the accuracy of prediction goes down. Using this kind of balancing techniques, you are keeping the prediction at a decent point, still in presence of very extreme imbalance. What about the model validation? Model validation for our purposes of prediction are based on resampling techniques. Basically, what you do is resample the database, and you try to reconstruct the probability function that you are interested in by empirically getting the shape of this kind of distribution. The problem is that when you want to do causal machine learning, we have discussed this previous session. What happened? You construct a propensity score using machine learning. Then you want to match, for instance, the lesser represented class, the is with other observation in the other class. And what happens if you want to enhance this resampling more than one observation in the other classes? The point is that you are the more you resample, the more units you match with your unit of interest, the higher the bias becomes. The simulation study we published, I think 1 year ago or something like that. The problem is that it's still you are still not getting a very good overlapping between the two samples, and you are losing a loss of accuracy in your estimates when you add more observations. So I think in this field there is a lot of work to be done to use how to derive causal interpretation in disease. As a possible roadmap of what we can do and what we will be doing in the next future is basically we need to add prior knowledge into the framework, and that was discussed already just before me. Bayesian models with shrinkage factor are there and can be used. I would say also that the importance of this is not a very statistical point, but I think the idea of getting more accurate information from the clinical point of view is important. If you have even a single observation which is not very well set or very derived, that can impact the model in your conclusions. And then, of course, you can also do some different. The idea, the very nice idea, is to use to mimic what is done in natural language processing. And what you do is basically you derive some feature outcome relationship for a general class of diseases, and then you anchor some features of your rare disease, the outcome according to some machine learning guided rules. So this is basically the idea. And I hope I answer your questions.
Well, considering my technical knowledge, it was more, but the fact that it's not something that it's impossible to do, it's because one of the problems was that if you have to just perform genome-wide association studies to identify modifiers, per se, you never have reached the statistical power. I think you need more than just basically what this is what you answered. Julie is raising her hand, please.
Yes, thank you. So it's a very, I really found this very interesting way of answering this question, and we are also, this is one of the kind of imperfect data set problem we have, the imbalanced data sets that and we are exploring this, and it maybe for you, but also for the panel, there are these possibilities with machine learning and the new techniques in deep learning to generate synthetic data or to do data augmentation. And sometimes when we talk about unbalanced data sets and so on, that's a solution as well that is proposed. And I'm having a bit of a, I never know if this is something that we should aim for in the genomic multiomic. Like, I feel like this is really difficult to do given that we don't have a lot of data, and then we want to kind of generate data, but it's so complex and we don't understand it. But maybe there's potential for these. So I just wanted to know the panel's opinion on these generative models in machine learning and their potential for data augmentation and for solving, in a way, those unbalanced data problems.
Yeah, I think this is exactly the point. In the severe imbalance, I think that augmentation techniques are really the only solution you can manage. You can have to manage the problem of using your information fully. And of course, this the problem of the way that after you augment the data set and in a different way, then how robust, let's say, robust your synthesis is with compared to the final model, and how your model, your conclusions are dependent on the model, because at that point the augmentation algorithm is somehow influencing the specific model you use for prediction. So it's not independent point. But this is definitely the only way we have. So we will go that way, everybody, I believe.
Amin, you raised your hand, right?
Yeah,
Yes.
so I wanted, in answer to Julie's comment, I just wanted to provide my own interpretation and experience with kind of similar situation where we are, for example, using adversarial networks to generate new single-cell transcriptomic data using models that have seen single-cell transcriptomic data. And our observation was that most of, for example, these kind of adversarial trainings rely on some hypothesis for trying to be, for example, confused, or trying to say that, oh, we cannot distinguish between the synthetic data and real data. And how much you can put, the amount of constraint that you put on that kind of critic that try to discriminate between these two would then influence the quality of the synthetic data you generate and its applicability. So for example, if your constraint is only at the distribution of the gene expression profile, you may not be able, your critic may get confused. But then if you try to get deeper into the biology and maybe learn transcriptional regulatory network out of those, that may have nothing to do with the biology of the real samples anymore. So the challenge to me there is that what is the end goal? And depending on how much domain knowledge you have, you may, basically, if you don't know where you are going to make mistake, you're going to go make mistakes that is going to be very detrimental. Basically, that's, I guess, what I'm trying to say. So, yeah, thank you.
Okay.
Thank you. Thank you very much for your answers.
Thank you very much. And if there are no, I will ask, then I will send an email to Professor Gregori, because we have a rare disease that is not so rare, so we could have a good amount, reasonably good amount of samples, and with rather detailed clinical data. And so I think this could be something that we could discuss working together with, if he agrees. Anyway, so now I have the next question is for actually, my question was, I didn't know what you were really speaking about, so I was asking whether these scoring system could be adapted for example, to neurodevelopmental disorder, because this is one of the issues in genetic diagnosis that is very hot, and because we know that we have families in which even if you run whole genome sequencing, you don't find anything, and we have the feeling that this is probably a polygenic disorder. So we find single variants, but none of them alone justifies the presence of the disease. If you for their specific characteristics, for example, you find many recessive mutations and no second mutation, so none of them is. But so my question was, can you adapt your models to do these kind of problems? But then I have also another specific question that came up listening to your presentation.
Okay, well, I can try to answer that. I am not an expert in neurodevelopmental disorders, so, but my understanding of it is that there was, well, there's different models, but some of it is also due to rare variation, as we discussed before, and sometimes even de novo mutations. So again, it's an issue of, sequencing the exome and then not finding anything, then thinking of, okay, maybe it's in the regulatory elements of some of the genes that we know of, and then we go look. And I've seen a lot of these talks where, you know, yes, they found for some families, but then there's always this bulk of, you know, unidentified, and so that's yeah.
50%.
Yeah, exactly. So depending on the study that but a large proportion. So I think that in that case, the way some people are approaching this is really trying to understand even more the variant prediction tools. So rather trying to infer or predict for an individual, you know, trying to find a score that, you know, predisposes the person, then it's more of a kind of the opposite side is variance and trying to predict their effect, and that's kind of another way of looking at things. But I think that your question is quite good, because I think one of the reasons why we are failing at these VUS, like the kind of variant of unknown significance, is that we are not looking at interactions between genes or variants, and even like from population genetics, I know about the importance of an haplotype, right? So even on the same gene, having multiple mutations maybe that are common and rare and so on, they can influence a lot. And we've seen this with RNA as well, like the structure and so on. It's a lot of mutations interacting together may have a big change. So I think right now, my take on everything, and might be a very simple way of getting out of this question, is the lack of consideration for interactions between genetic loci, maybe in cis, so in the gene itself with regulatory elements and so on, and maybe in trans, you know, with other genes. And, you know, statisticians don't like me when I say that, because of course it's complex to do and it's the limitation. But this is, yeah, this is what I can say on this.
If it were easy, you weren't here. I mean, please let us hear your opinion, because
thank you. No, it's a different question. So if you want to discuss it, I can wait until.
No, well, I had another question, which is much more population specific, and this came out of your presentation. And you were talking, if I understood correctly, to have ways to take away ethnic specific variants for your analysis. But are you sure it's a good idea? I'm talking. I was thinking of the sickle cell anemia mutation, which by alone it can predict ethnicity. You find an individual with the sickle cell anemia in heterozygosis, you can be not absolutely sure, but 95% sure just by that single variant that is of African descent, because in Europeans it's 1 in 16,000, and in Africa is 1 in 10 to 1 in 5, depends from the area. So you did just one variant. But this is a variant, it's a common variant with a very strong, because this is a semi-dominant disorder. So are you sure that eliminating these variants is a good idea? And same thing with the fibrosis mutation or other.
Right. So it's not the case. Like the adversarial, the domain adversarial neural networks that we're thinking about would not eliminate. That's what we don't want, of course. And you know, there's a lot of these. So you're mentioning that, and there might be. So there's two things in the ethnicity questions, right? One thing is that we don't want to be predicting based on variants that have nothing to do with the phenotype, just because they are associated with a certain ethnicity that has a higher predisposition or prevalence of a disease. That's what, honestly, like for looking at these scores, the polygenic risk scores that take all the genome, they are biased towards strange things in the population they're trained on that are associated with some factors and associated with population stratification. And so an example for cardiovascular disease, for example, in the UK Biobank, if you use those scores and you look at the stratification, all of a sudden you find that different regions have different predisposition, and it correlates with, for example, deprivation index. So, you know, more poverty, so on, but also with the structure of the population. So then it borrows information from, you know, who lives there in those areas, what genetics they have, and uses that because it gives a better predictive power, but it has nothing to do with the biology. It's just the association, and this is, so my point with this, and of course that's what we don't want, to be excluding variants that are differentiated between populations to predict a trait, because this other aspect is true, as you just mentioned with this great example, is that sometimes you have variants that in different populations will influence the phenotype in different ways, and you want to keep them. But what you don't want is a general approach that is biasing towards ethnicity. And so, of course, it's in the case of complex diseases with many genetic variants within. So, because if you do that with the sickle cell disease allele, just that, for sure you won't be able to distinguish, because this is the cause of the disease. But when you have a lot of genes, the idea is to try to make your approach sensitive to the disease, but without it being able necessarily to predict the ethnicity of the participant, such that it's not taking out the ethnicity of the participant that is still in its genetic background, but the bias towards predicting something that would be related to where, you know, the person lives, or the person's you know, ethnicity and other things correlated with it. It's exactly, so I guess what I'm trying to say, it's in practice, it's exactly trying not to remove SNPs, but weighting them in a way that they're not, the network is really finding the association with the disease and not with the ethnicity that might correlate with the disease.
Yeah,
I don't know if I'm
it's clear. I don't understand how you do it, but
well, and to be honest, this is under development, and I'm not sure it will work, but this is the way, you know, in genome-wide association studies, they just put the variable, like the PCAs or the ethnicity, and in the model, and they correct for this. And so this is a way that I think with these neural network models, we can try to do this. And maybe there's other ways, and I'll be interested in knowing more if any of you have, but we need to correct for a lot of things, and, you know, ethnicity, but sex, same. If sometimes we have those problems where genetic risk scores doesn't perform as well in male and female, and in a sense, we need to make sure that the model, in a way, can understand it's a female and predict the right score for that one without, you know, just predicting female in a sense.
Exactly. Thank you very much. Amin, please go ahead. Sorry if
No. My question is actually related, and you can think of it as also a follow-up to your question. So it's related to, you know, domain adversarial training for domain adaptation, basically the algorithm DAN that you used. DAN, for example, which was originally developed for computer vision, originally focuses on a discriminator that tries to confuse a binary classifier, right? So a lot of confounders or a lot of variables that we usually have in our field is, however, non-binary. And when you switch that binary classifier with a multi-class classification, training of that domain adapter or discriminator becomes very difficult, again, for the data imbalance. So I wanted to know your opinion, because we couldn't get much success out of that. Like, we tried DAN, we tried ADA, we tried a lot of, and it wasn't it was actually hurting us more than helping us in our particular application, even not going to multi-class classification. But when we go to multi-class classification, that becomes even more challenging. So I wanted to know more about your experience with that and whether that was something that gave you success in your use cases.
Yeah, so I agree, and this might be one of the reasons why we're not succeeding too much. We have too many classes, and we try to make it more simple and so on, with just doing something very cleaner with just European Africans, and see if we can get some good results for that. I guess, and we did male female, so that worked quite well. And so for some applications, in at least making the full network not being able to distinguish, because we were trying stuff on the X chromosome, and so we wanted it to be really the genetics of it, and in those more simple cases we had some success and some, and I think I relate a lot with what you're saying. It's not perfect, and we keep working on it, but we also have the problem that it's not either perfect for the other side of things, predicting the phenotype in itself. So
absolutely, I completely agree. I think it would be great if later on we can get in touch and talk a little bit about this.
Yes, absolutely.
Thank you very much, Lee. And now is for Professor Sperduti. And again, as I mentioned earlier, you have to forgive me because I was trained as a pediatrician and I ended up in genetics. So I will ask disease questions that are relevant for fatally for the disorder I deal with every day. And when I tried to read and understand your papers, because my knowledge certainly was very below the par to grasp that. But understanding for your presentation and from at least from the abstracts of your paper, I found very interesting your approach to find these disease gene associations for relatively large course of patients. And this is the whole point of what you are doing, and the current line of research for complex research. But is it, and this is similar to what I asked to Professor Dario Gregori, what about novel phenotypes in few patients? Is this applicable to it, or it's just a waste of time because you be overfitting data and whatever? I mean, or trying to address it. I don't know.
Okay. Yeah, thanks for the question. Of course, everyone has his own knowledge. So my knowledge is more on the machine learning part. You have knowledge on genetics and genomics. So I think it's fair, and we are here to try to discuss how we can interact to get a better solution to problems that are currently unsolved. So one advantage of kernels is the fact that basically they are less sensitive to the number of examples, because in a sense the kernel, as I was mentioning in my presentation, is giving a measure of similarity. And so one standard approach is not using learning at all for prioritizing the genes. And the idea is just to define similarities measures that are handcrafted. And then basically the system is combining the similarity according to what is the kind of database that is used in order to rank the genes according to the positive examples, okay?
Yes.
Okay, so the approach that we were using, Scuba, is adding on top of that a degree of adaptation, because the kernel, that we have been using, they are in a sense predefined. They are actually using topological features, but it's basically a similarity based on how far in the graph the nodes are, basically. I'm simplifying that, but basically this is the idea. And then the machine learning part is trying to understand how much credit should be put on each of these kernels. And so the idea was to recombine these similarity measures in such a way to be more consistent with the few genes that have been considered to be positive. So from a general point of view, I would say you can also use a single gene. Of course, this approach is not applicable if you have a monogenic disease, because you have just one. So, the assumption.
This is the point of what I was asking to Julie, because we have the impressions that many of these situations in which we don't find a clear mutation are actually polygenic. And this is so we may have a common biochemical phenotype or a biochemical trace, but would it be possible? From what I'm understanding for you, what you're saying is that it could be possible to investigate whether the different patients have similar genetic signatures. Am I understood correctly?
So actually, in principle, you could also use just one gene or few genes and then train the system to weight in a proper way the different similarity measure, and so you can still get a ranking. Of course, the more positive genes you have, the better would be the confidence in your ranking. So I'm not saying that it is going to work very well, but since basically the training examples for us are the genes. And so what we do is just trying to, okay, genes in our setting are positive or unlabeled. Among the unlabeled, you may have positive or negative. And so the trick that we were using was to assume that in the unlabeled, the entropy was maximized because you have no knowledge about the class of the genes. And so making this assumption, we were able to perform learning only considering the kernel value, so the similarity value between the positive and the unlabeled. So this means that it's actually possible just to use one gene as positive, and then you can use this information to combine the different similarity measures coming from the different databases, and then you can still get a ranking that can make sense. At least you do have some starting point for your investigation. And then, about the explainability, this is another issue, because then it's very hard to extract from this kind of systems an explanation. Well, you can say, okay, since the kernel is computing the distance more or less from the two nodes, then you can find an explanation working very hard, but there is a lot of contribution from the human expert to extract the explanation. So I think that the nice thing of kernel methods is that they are easier to use when you have a few examples. And even because they are more regularized, as Professor Gregori was mentioning, the importance of regularization. And so at least for this specific task, they can be quite useful. But then I'm sure that deep learning approaches can really improve the situation. If you look at the alpha folding network, that was amazing what they were able to do. And so I think that sooner or later we will end up with better solution in general.
Thank you very much. And if no one wants to add anything, I'll ask my question for Jerome. And and I have to make a premise for our Canadians. And the issue of RNA therapeutics is extremely hot in Italy right now, because it's one of the fields of research funded through these new funds for recovery funds for the pandemics. So it's very at this point it's one of the hottest issues that is currently being discussed, and there are literally hundreds of millions of dollars in funding at stakes on these issues. So I was very interested in your talk. And could you? I mean, my question was similar. We have examples of RNA targeting of specific RNAs for therapy. For example, spinal muscular atrophy is one of the fields where you can target specific RNA and either promote splicing or whatever. But what are the other targets that could be therapeutic targets at this point of the other mRNA species? Because you showed us it's not only mRNA, tRNA, and ribosomal RNA.
Well, I mean, to some extent, I mean, it's a bit an open question, because we haven't discovered all functions of RNA yet. They're really literally involved in any process in the cells, different degrees, of course, but they're playing so many different roles that really a very large opportunities for many different applications. One example I like, and I'm trying also to push, is of course ribose switches. So ribose switches are elements that you find usually in a promoter region of genes in bacteria. And we have a couple of examples for eukaryotes, but not for mostly. I mean, not for mostly for plants, but not really for human, or at least not that we know now. But that's very interesting target, for instance, for designing new antibacterial drugs.
Yeah. Okay.
Because we found them primarily in bacteria, so that makes them a good target to avoid off-target effects, of course. And so Merck a while ago, I don't remember the name of the drug on top of my head now, but I mean, basically tried to develop a drug to target one ribose switch to make an antibiotic. Didn't work. I think it stopped in phase 2, but still it opened an interesting area, and now more and more research programs are trying to target these elements. So it's very promising from my perspective, because as we all know, AMR, so antimicrobial resistance, is one of the major issues in upcoming years, because it touches everything literally. Ribose switch is really the example I really like here, because it shows that it's a highly structural element, and they're really relying on a binding to small molecules to function properly, and that makes it is a promising target. But of course, after we have so many, as I said, we can also imagine like it's the snow RNAs, any other RNAs, the microRNAs, of course, are already one target that is already very much studied. And everything that every structural element involved in gene regulation is a good target in general, indeed. After the main question will be that do we want to target this element with a small molecule that makes it easier perhaps for delivery, but has potentially much more off-target effect, or do we try to design other siRNA therapy, for instance? That's a question that remains open, but it's I think we're just scratching the surface now, and it's this is why it makes the thing so appealing.
Can I ask another question? Is there anybody that wants to ask? Because I have 2 other questions. I mean, please, because I'm the moderator. I'm not supposed to ask too many questions.
Yeah, thank you. Jerome, I have a question. In your presentation, you talked about graph convolutional network and networks. And if I understand correctly, that was to learn representations from secondary RNA structures. At the same time, you have developed this very nice catalog, Carnival, that captures some other aspects of the secondary architecture, and structure. And I was wondering, either as an alternative to graph convolutional network or as an augmentation, the representation or what you have learned in the maximal common subgraphs in the Carnival can be used again as an alternative to graph convolutional networks, or to augment it for whatever downstream task that you have.
Well, it's a very good question, and to be fair, for now, it's even Vernal for us is very new. So graph convolutional network seems to be the best approach, and the one that it was because graph machine learning in general is relatively new, even in the field of machine learning. So it's we didn't want to be too much adventurous, right, in these fields, and we have a bit more understanding about it does. I'm pretty sure there's many technical aspects that can be improved in a framework.
The reason I'm bringing it up is that exactly what you're saying is that graph convolutional networks, while they're maybe very helpful, they also come with their challenges of training them, etc., etc. But given, for example, Carnival, at least if nothing else, that may help make them interpretable, like by understanding what are the representations. Again, I don't have like a very solid idea, but I'm just thinking that this could be an interesting avenue.
Yeah, absolutely. I mean, actually, to be fair, so Carnival, it was more like a classical maximum subisomorphism problem we try to solve with combinatorial algorithm. And then after we moved to this more fuzzy detection of different class of motifs. And I think the strength of this is rather to, exactly as I was suggesting, comparing both, to use the knowledge we have on Carnival to have a better interpretation of the result found with Vernal. So we've been able to see that interestingly, the class were relatively similar. So Carnival the database was enabling us to look at very specific motifs and better understanding them, because we're sure that conservation of structure have a better interpretation of Vernal data. And eventually, as I was suggesting, maybe some result to get with Carnival could be used to seed the graph neural network technique that we have. But it's a very open field, yeah, indeed. I would say that
we have some similar kind of challenges in kind of new drug design, like because we can also look at the graph structure of the drugs and stuff. So that's some of it. Yeah.
Alessandro?
Yeah, this is just a comment on graph convolution neural network, because we are actively researching on that, and it's very well known that there is this over-smoothing problem for graph neural network that does not allow to have too deep networks. And so there is a lot of work that are trying to go around that. And we did with our group some experiments using still convolutional approach based on Laplacian transformations, where the idea was to try to have not stacking of layers, but having a priori power exponentiation of the adjacency matrix in order to have all the range of distances in the convolution, and then applying on top a standard, more or less a standard deep network. And this seems to be a better performance, but there are other works in the literature where they are actually addressing this kind of problem. When in my presentation, I was mentioning the fact that the problem when you use graphs is that many techniques are actually using global information. I meant something like that, because the problem of convolution graph networks is that they look at the whole neighborhood, and then they try to combine the neighborhood, but using an approach that is was criticized by Julie when the task is not stationary, no? So you are actually applying the same function to all the nodes in your graph. But this may not be the case. And also what you would like to is maybe using some attention mechanism to select maybe a subset of the neighborhoods. And what I think is also very important, and I try to explain in my presentation, is that very often maybe you have multiple causes for the effect. So which means that maybe you need to look at a small portion of the graph in some position and then some other subgraph that is far away from the previous one. And using convolutional graph neural networks, this is very difficult to do because you basically grow your knowledge from local to global by stacking several layers. And this is what is giving you then the oversmoothing effect. So in principle, the convolution idea is nice because it's overcoming the problem with isomorphism, because basically you are sure that you have no problem with different representation of the same graph. But this is also bringing with itself this problem that you are actually mixing everything together. And so my position is that we should try to have a different approach where basically try to select in a smart way, maybe learning what are the topological features which are really important.
Thank you very much. I have, if nobody has, because then I think I wanted to ask you a general comment on data overfitting, but I think we are way over the limits, and so I think this is not something at least. But I had two very brief questions for Jerome, and one is, and this is strictly related to our fields of research. Now, we study among them mitochondrial disease, and as I was mentioning earlier, we have mutations affecting the mitochondrial tRNA genes. And so one is a very trivial question. We have two novel mutations, one in tRNA glycine, and the other is I think it's tRNA histidine. And I mean, would you be interested in modeling the mutation in using your tools to see what would be the structural consequences? But this is okay, clear point. The other point is instead probably more general. Some of these tRNA mutations cause stable RNAs that tRNAs that are degraded. Now, would it be possible to design drugs that would act as chaperones to refold these RNA, similar to what's done with chaperone therapy, for example, for enzymatic disorders? So is it possible to, by knowing the structure of the RNA, knowing the structure of the altered tRNA, to design specific drugs that would help in refolding the molecule, or because the other approach would be just take 2,000 compounds, see which would restore cellular respiration in cells, but this is a brute force approach that sometimes is not. But the rational drug design approach would be possible in such, according to your experience, or it's something that, besides, you have in the field one of the most top world experts of mitochondrial disorders. So if you want, also I can indicate who you could collaborate on this there is Eric Schubridge. So if you want a collaborator in Canada, you already have it in the institution.
Yeah, well, what I say, so, well, first, tRNA is always tricky, right? Because there are chemical modification that helps the folding, and that's always make more complicated. And most of the folding algorithm actually do not necessarily handle that very well. I mean, not at least as the unmodified, when there's no modified nucleotide. Yeah, it's still possible to, I mean, there's multiple different analyses that can be done indeed. I would also every time go against a single answer to this, because there's so many different type of analyses that can be done, like try to first to map the sequence and the non-structure, see where it can be destabilized, to try to run some MD simulation. After we can look at also how mutation could impact indeed the structural ensembles. And that can be done with more classical tools, such as the one I was presenting. So of course, there'll be more like accumulation of evidences that can be here to support eventually some experiment that can be done on the folding of tRNA itself. But just to point out that tRNA is always a tricky to study because of the chemical modifications. Now, I think we're a bit far now to, I mean, we're not there yet, or at least I don't know, I haven't seen an example of that, of designing drugs could help the folding. Specifically because, I mean, there's so many RNA are quite flexible. There's a lot of interaction with water, so that, and other ions around that can drastically affect the folding of the molecule. Eventually, we could imagine, I mean, as theoretically possible indeed, to try to see if some additions of small molecule could help the folding in the good structure rather than the others. That's thing that we can try to, that could be explored. But that would need to be indeed after validated with experiment behind and incorporating the feedback. And there will be a full research project, basically. That would take several years, I guess, yeah.
Thank you very much. I think that it's 6 p.m. here in Italy, and I guess it's noon in Montreal. So I think it's about time. I would say here, if we could go out for a spritz, so I think this has been a fruitful discussion, and I personally learned lots of interesting things, and I had I hope that also you have enjoyed the discussion. I don't know if there are any additional comments. If one wants to make anything else to the discussion. On my side, to draw conclusions for what we discussed today is very difficult, because we dealt with so many different issues, and which are, again, as I was saying, far from my own scientific expertise. That, I mean, it would be probably, but I think that this has been an interesting confrontation, and also that hopefully that some collaboration could originate from what we discussed here today. And I want also to thank all the people who attended this session, and the people who are watching us on YouTube, and thank again Professor Gerosa and the Consortium CORIS for organizing this event. If there are any other comments, I thank you all, and I hope to see you soon, and possibly that next time we do this in person. And so rather than that's something, because Zoom or Meet are good, but are really instruments, but there's nothing like getting together to exchange ideas. And so it has been a pleasure, and thank you very much.
Thank you, everyone.
Thank you.
Thank you, everybody.
Thank you. Bye.
Bye.